Am I audible now? I hope I am audible now. Good afternoon.
Yes.
Welcome to PALS Theory to Practice lecture. I am Robin from PALS Program Management Office. So I'll be the virtual host for this session. Before getting into the session, the participants should follow ground rules. So all are stay muted. Please record your attendance in the attendance form, which will be posted in the chat box. Suppose if you are not already registered, so please register in the registration form as well as the attendance form. So you will have the flash quiz at the end of the session. So suppose if you have any questions, you will have open-ended questions at the end of the session. So we will share the feedback as well as the online quiz at the end of the session. So till now, stay connected. So before getting into the session, I would request Mr. Ram to give an intro about PALS as well as the speaker's profile. Ram, sir.
Thank you, Robin. Am I audible?
Yes, sir.
Okay. So this is the first of this year's PALS Theory to Practice lectures. I will not go about all the offerings of PALS. This is a student event. And it is the first theory to practice. And today, it's based on cancer therapy. And then it's a subject which has been researched. And the researchers have got the Nobel Prize for this subject. And it has also come into the clinics already. So that's why both the theory and the practice today's speaker will cover. I'm happy to start the proceedings with a very warm good afternoon to the students and faculty, the PALS institutions and my colleagues in PALS. I hope you're all energetic and alert as the topic is of great importance and interest to us as human beings. Cancer therapy by inhibition of immune checkpoints. Now, about today's session, a little bit. During the EC orientation conducted a few weeks ago, there was a remark that so far we did not have any session on biotechnology. We have been having mostly on core group, mechanical and other digital areas. So PALS has responded immediately to the request. And today we have amidst us a professor from IIT Madras, Dr. D. Karunakaran, who is an eminent researcher in cancer with many laurels to his credit. Dr. Karunakaran has been in IIT Madras since 2006. He joined as professor in the Department of Biotechnology on 1st September 2006. He was nominated as the Institute Professor Chair in May 2018 and continues to teach at IIT, designated as Professor Emeritus. Dr. Karunakaran obtained his MSc and MPhil degrees in biochemistry from the University of Madras in 1979 and 1984 respectively, and later obtained his PhD in biochemistry from C. Krishnadevaraya University in 1991. He started his teaching career as a lecturer in biochemistry at the PSG College of Arts and Science, Coimbatore in August 1979, where he taught until 1992. During that time, we have known him personally to be a soft-spoken and diligent teacher who was very popular among his students at PSG. After that, he went on to do his postdoctoral research training at the Weizmann Institute of Science, Israel. And then again, he moved to MD Anderson Cancer Center in Houston, in USA, where he met he was associated with Dr. James Allison, who later got the Nobel Prize in 2018. Dr. Karunakaran then, prior to joining IIT Madras, worked as senior scientist at the Rajiv Gandhi Center for Biotechnology, Trivandrum, from 1998 to 2006, and from 2006 he has been with IIT Madras. Professor Karunakaran's research interest is in the field of cancer biology. He has received the C.R. Krishnamurthy Lecture Award for the year 2003 by the Society of Biological Chemists, India, and the Kinariwala Lifetime Research Award for Cancer Research, Gujarat Cancer Research Institute in 2018. He was a visiting research fellow between 2008 and 10 at the University of New South Wales, Sydney, Australia. He has authored more than 80 original research papers and is an inventor with 2 patents. He has trained 16 youngsters at the postdoctoral level, guided 15 PhD and 5 MS students. Today's session is based on the research of James Allison and Tasuku Kohonjo of Kyoto University in Japan, who won the 2018 Nobel Prize. Briefly about the topic, we all know that the ultimate aim of immunotherapy is to boost the immune system. Our body's natural immune systems destroy cancer cells every day, but this is impeded by checkpoints that act as brakes. So this research, introduction of T cell targeted immunomodulators, block such checkpoints, and then it's able to help the treatment. And the more on the subject, I will leave it to Dr. Karunakaran. So we are immensely fortunate to have Dr. Karunakaran inaugurating the T2P session of this year with the subject on cancer therapy. So I hand over to Dr. Karunakaran. Thank you. Over to Dr. Karunakaran.
Thank you, Ram, for the nice introduction. First of all, let me thank the organizers for having invited me. It's always a pleasure to address students and other academic people to share whatever I know. So without much ado, let us dwell into the topic. How do I show the full screen? Is there any? I mean,
you have the slideshow mode would be there.
Yeah, I have put it. I think I'll have to remove this. This some. Okay. I found out myself. Okay, good. So the topic is cancer therapy by inhibition of immune checkpoints. Let me clarify. I'm a cancer biologist. I'm not really an immunologist. But I chose this topic mainly prompted by Ram's ideas. I mean, like the topic that he told me, that is theory to practice. So I was just looking for something that has come into the practice. People start with some basic research. All of us are basic researchers. I haven't done something that has just come into the clinic or something like that. I'm not claiming that also. But what happens is such a process is usually lengthier, and it takes efforts from practically hundreds of scientists, and then you will have to finally apply, and then it comes into the clinic and people get benefited. Because you see, both the basic science and the translational work are two sides of the same coin. We cannot debate which one is more important or less important and so on. Somebody said, Max Planck said, there must be science to apply. So there must be something, it's like a foundation you must have. So the theory and practice, they go together. One doesn't live without the other. So that is the whole idea. That's why I chose this topic, because this topic is about basic immunology, but it has found its way into the clinic. And as Ram mentioned, the people got Nobel Prize as well. Yes. So these are the two people, James Allison and Honjo. They discovered kind of, this PD1 was discovered by one of them, but the second one, CTLA4, was not discovered by them, but the mechanism of action was discovered by Allison, because of which they got the Nobel Prize. Today we'll be talking about their discoveries. I will go to the next slide. All of you know, as Ram also mentioned, what is immune system. Basically, everybody knows that immune system protects us. Today in COVID times, I don't have to explain. Everybody, even who are not scientists, know something like vaccination to protect us from being affected by virus or bacteria and so on. So we know that our immune system protects. And you also know, the younger you are, you have a better immune system. The older you are, your immune system becomes weakened. So these are all some general concepts that you have to understand. What is the main function of the immune system? It is basically, it is the system that distinguishes what are the molecules that you have in your system and what is coming into your system from outside, that is foreign material, virus, bacteria. This is not, I mean, this is present also in our system, but something that is unique, that is not present in our system, and it affects us, infects us, then that is being recognized by the immune system. And basically, most of the times, they are successful in destroying them so that you and I do not get any serious disease in our day-to-day life. So that is the function. Now, one of the major ideas about the immune system and tumor or the cancer and tumor I am using interchangeably. So one of the ideas is that how do these immune system protect us from getting cancer? I mean, like, it prevents us from getting cancer. So everybody will say, yes, yeah, probably immune system gives us a protection, but how? Scientifically, the question is, the tumor cells, what are cancer cells? Well, let us begin from there. Cancer cells, everybody thinks that proliferating cells are cancer cells. This is a very wrong idea, because you see, there are many cells that proliferate even under physiological conditions. Breast is one example. Female breast grows, the cells grow. But there are many other examples where a mere proliferation is not cancer. So proliferation is part of that. Cancer cells, of course, grow, they proliferate, but that is not the only identity. There are many things. Similarly, you should also understand in biology certain basic concepts I will tell, because that will help you to understand this whole lecture. So I am beginning with certain simple things. Sometimes it may be, for those who are already aware of it, it may be a little bit boring, but for those who do not know, then it will be of interest. For example, all of you must have heard about human genome. How many genes are there in the human cell? What is a genome? Genome is the total genetic content of a cell in this case. If it is a human cell, in one human cell, what is the total genetic content? That is its genome. So how many genes are there? People say about 20 to 30,000 genes are there in the human genome. That's what people have researched and found out. Now you may ask, sir, cancer also is related to genes. And so how many genes are involved in the causation of cancer? You will be surprised. There are only about, I told you, about 20 to 30,000 genes, but about 300 genes are associated with cancer. So you may think only 10% of the genes are associated, then how is it becoming such a serious disease? Who does not know about cancer? Some relative, some friend, somebody known to you in some way or other has been affected by this unfortunate disease. And that prompted a doctor to call this as emperor of all maladies. And he wrote a book. You can read that, Siddharth Mukherjee. So he wrote a book on this. So to call this as emperor of all maladies, what prompted that is because you see it's very common, and you see people getting cancer, and you also know that it cannot be cured so easily. That is the notion people have. In fact, there is an interesting story about the Cancer Institute in Madras. When it was started, Dr. Shanta used to recollect that story. That is, it's not story actually, it's what happened. When she and other people together wanted to start this hospital, one minister at that time asked her, why do you need a hospital for a cancer patient? After all, they are going to die. So this was the notion. So, but today it is not like that. Cancer is treatable, and especially if it is detected early. Now, I will not distract too much. Now, I told you there are about 300 genes, and these genes interact with each other, especially the proteins, they interact with each other. So it is a chain reaction. It is not that just only 300 genes are associated. Because of their interaction with other genes and other proteins, ultimately many genes are involved. It's not, what I want you to understand is that it's not a single gene. Another interesting thing is, you must have heard of oncogenes, which are the growth promoting genes, which are the cancer causing genes. Where do they come from? They are from your normal gene. Whatever genes you have, they are tinkered, mutated, and then they turn into oncogene. So that raises to this question that this slide shows you. Tumor cells arise from normal cells. So the tumor cells are not alien cells. They are there in your system. They were once upon a time, they were normal cells. Due to something, they have been mutated or converted into cancer cells. So the question comes now to immunology. If it is a normal cell, if it is already present in the system, how the immune system will recognize? Because immune system is not supposed to recognize what is already present in your system. So that is the basic question that people had and could not believe that the immune system could protect us from cancer. I am talking about history. Some years back, some 40, 50 years back, this was the notion. People were refusing to believe that immune system could protect us. Now we know that it does protect. I will give you some examples. Now, as I told you, I am not an immunologist, and it will be difficult for me to explain everything in immunology, but it becomes important because the topic is something that the people got Nobel Prize for immunotherapy, and without telling something about immunology, it will be very difficult to follow the findings of these scientists. So I will just touch upon, I will give you just an overview. So as I told you, the microbes, viruses, bacteria, and other foreign materials, you have something inherent in you, something that is naturally present in you that protects certain cells. For example, the first line of protection is your skin itself. Skin doesn't allow many foreign particles or viruses, bacteria to go inside. It acts as a layer of protection. So similarly, you have other protective things in your system. And apart from that, several molecules and cells are there to help you. What is present inherently in you, naturally in you, is called innate immunity. That is, it's already present. It is not formed after something comes in contact with you. Before something comes into contact with you, it is already present as a protection. What are those things? One is this epithelial barrier. Just now I told you skin, for example. What are known as phagocytes. Phago means eating. So they will eat the foreign material and digest it and ultimately kill the bacteria and viruses. So ultimately, you are not getting any disease, you are not getting any infections because of this in the normal day-to-day life. And there is another one that you have to understand is what is called NK cells, that is natural killer cells. Let me take the pointer, just 1 minute. Yes, I hope you are able to see. This is what is called NK cells, natural killer cells. Now, this is the immediate reaction. When you have some infection, virus, bacteria, and all that, within hours, within a few hours, let us say 6 to 12 hours, within that, you try to kill those organisms, those microorganisms, because they are dangerous to you, so you kill them. So this is happening immediately. So that is taken care by innate immunity. By that count, it is non-specific. By which what I mean is, it doesn't matter whether it is bacteria, virus, or this bacteria, that bacteria, doesn't matter. Anything foreign will be destroyed by these cells. But for everything here, there is some limit, threshold. The body cannot withstand everything. You know, if you dump everything, then there is a limit. There is a limit for everything. And another interesting concept that I want to share with you in biology is that, you see, what is a living organism? If you say something as a living organism, it may be something, a bacterium, or a virus is controversial, but bacterium or some other microbes or animals, plants, everything you call as living organism. So what is the basic idea? The fundamental physiological and functional unit, structural and functional unit of living organisms is a cell. So by definition, although the definition, I'm simplifying that, by definition, something if you want to call, at least call as a living organism, at least it should have one cell. So such organisms, we call them as unicellular organisms. If you have more cells, you call them as multicellular organisms. Why I am telling these cells have the machinery to fight also for the immune system. There is another important thing in the about the biology of the cells and living organisms. For the cells to function, whether to grow or proliferate or even to die or move, migrate, differentiate, or to communicate, chemical communications or neuronal conduction, muscular contraction, any work you can imagine that your living cells do, they require a signal. So these are all the important things that you should know before I begin the lecture. Now, the cells will function only if there is a signal. This, for example, here, these microbes, foreign materials, they will give a signal to these cells. So immediately the signals are picked up and they recognize this and then they kill them, for example. Let us take like that. So like that, there must be signals. Again, interestingly, in biology, you should also understand, all of you know that Newton's third law. For every action, there is an equal and opposite reaction, they say. But here in biology, it may not be true exactly. It may not be equal. It may be unequal or semi-equal or something like that. It can have gradation, graded response. Why I am saying like that? For every biomolecule you can imagine, you will have something opposite. For example, we study what is known as apoptosis, a programmed cell death, which means there are some molecules which can induce cell death. You call them as pro-apoptotic molecules. Exactly opposite, which will prevent the cell death, you have certain molecules. You call them as anti-apoptotics. Similarly, I can go on telling about these things. If there is an inducer, there is an inhibitor. If there is something that activates, then there is an inhibitor. Now, this concept is very important to understand. So when you inhibit an inhibitor, you activate. So this concept also has to be understood because this has tremendous relevance to the lecture that I'm going to give now. Here, we are talking about the immune system. I told you that there is an innate immunity which takes care of the immediate needs. And after some days, the immune system tries to recognize that and tries to develop certain molecules based on the structure of the molecule that it has encountered and produces what are known as antibodies. These are produced by B lymphocytes, and you have certain, specifically the antibody secreting cells are called plasma cells, which are nothing but some kind of matured B cells. And there are also what are known as T lymphocytes. You should know this. They have to be these T lymphocytes, the so-called T cells, they have to be activated. They become effector T cells. And these are important to kill the foreign infected organisms or whatever. I will just show you that. Such killing T cells are called cytotoxic T lymphocytes. So they will kill the organisms. Similarly, the antibodies also will neutralize the foreign materials, so the so-called antigens. So antigens will come and bind with this antibody and ultimately it could be destroyed. For simplicity, I am saying like that. So the innate immunity takes care of all the foreign proteins, viruses, bacteria, parasites, fungi, or any kind of infection. The innate immunity will try to take care of it. But sometimes it may be that you have so much of viruses or the threshold crosses, then also the time passes, and if there is a persistent infection, then adaptive immunity also comes into the picture. Together, this innate immunity and adaptive immunity They take care of us, protect us, and so on. When the normal cells take the journey of becoming a cancer cell, it is a long journey. So it takes several mutations and other parameters to get cancer. So it takes it's many years also in some cases. You may ask, our children also are getting cancer? Yes, yes, it is there. But the time duration, it definitely takes some time for anyone to get cancer. It is said to be something like an evolutionary process. So there must be a lot of mutations that have to take place. So what happens is, initially, the cancer cells, the normal cells that are going to become cancer cells are recognized by the immune system and are destroyed. And then some equilibrium is reached. Sometimes the cells become resistant, and then they turn out to be ultimately, once the threshold is broken, then they become cancer cells. So there is a constant fight between the cancer cells or the so-called neoplastic cells, which are going to become cancer cells, and the immune cells. And so they fight with each other. Whoever wins the war will have a different end. If immune cells win the war, then you don't get cancer. If the immune cells are defeated, then you get cancer. So you can take it like that for simplicity, for an understanding, which is also true scientifically. Now, I will not give this overview again because it is too many things are there. Just I want you to understand that there are antibodies, there are this cytotoxic T cells, that is CTLs, cytotoxic T lymphocytes as they are called, CTLs, which will kill the foreign material. So that is very, very important for us to understand. Of course, innate immunity, I already told you. Now, the antibodies, because the talk is about antibodies, so I have to tell something about antibodies. Antibodies are produced by activated B lymphocytes. As I told you, they are called plasma cells. They look like this. Structurally, they look like this Y-shaped structure. There is a heavy chain, light chain, and a body, and so on, constant region, variable region, and all that. So it looks like that a pair of heavy chains and a pair of light chains joined by so-called disulfide bridges, and so on. So the foreign one that comes and binds is known as an antigen. So the antibody naturally binds with the antigen. So it has got antigen binding domains. So up to that, you understand. Now, I will come back to this question. Sir, how do you say that you are saying immune system protects us? How do you know that? There are some evidences. What are the evidences? There is also the other question that I asked. Sir, if normal cells are becoming cancer cells, normal cells are not supposed to be recognized by the immune system. Then why cancer cells are recognized by the immune system? What happens is, a few proteins, they become structurally novel. That is, they get mutated. Now they attain a different structure. Now they look like a foreign molecule to the immune system. That is why cancers like the immune system is able to recognize them as foreign or antigenic scientifically. And so cancer cells do have certain antigens, and they can be recognized by the immune system and they can be killed. Only thing is, then you may ask why we are getting cancer. As I told you, it is a war between immune cells and the normal cells, or the so-called pre-neoplastic cells, the cells that are going to become cancer cells. If the war is won by the cancer cells, you get cancer, for simplicity. There is another thing that you have to understand. For example, there are certain oncogenes. In this case, I have just put it as RAS oncogene. RAS oncogene, RAS is normal, but if it becomes oncogenic, then it undergoes some change, so that can be recognized by the mutated RAS. Can be recognized by the immune system. Similarly, there is another apoptotic protein called P53. It's a tumor suppressor. There is also oncogenes. As I told you, for everything, there is an opposite thing. Oncogenes, they growth promote. They produce cancer. Tumor suppressor genes, they are doing the opposite. They will inhibit the cancer cells and thereby prevent you from getting cancer. So there are again opposite things. In biology, everywhere you go, in all the systems, you will read this opposing partners, something like Jekyll and Hyde. There will be good and bad. So what happens in the end is the compromise. Somebody wins, somebody loses. And the same thing, same person may not win all the time. Sometimes oncogenes win, sometimes the tumor suppressors win. So that's how you have to understand. Then it becomes easier for you to understand. Like I told you, suppose you suppress a tumor suppressor, you will be activating the process of cancer formation. So that is also another important thing. So this is why cancer cells are being recognized by the immune system. Now I will come back to that other question. Sir, you told that the immune system protects. How do you know? What is the evidence? There are many evidences, but I will give you some simple examples. For example, immune system protects the body from virally induced cancers. Don't think that cancers are induced by viruses. They are only 15% of, if you have 100 types of cancers, only 15 of them are viral origin. So it's only a small percentage of cancers are due to viral origin. All others are not well. Similarly, another wrong notion about cancer, especially breast cancer, people are thinking it is always hereditary. It's not like that. Only 5% of the patients have the hereditary. This one, all others are what we call as sporadic. That means non-hereditary reasons. Hereditary is there. I am not saying it is not hereditary at all. Only a small percentage is hereditary is what I am trying to say. In the case of breast cancer, for example, 95% is not hereditary. Only 5% is hereditary. So that may be surprising to some people, but that's another thing. So immune system protects us, for example, virally induced. And see, for example, if people are already having immunosuppressive drugs, they are prone to getting cancer. For example, AIDS patients, their immune system is suppressed. Everybody knows AIDS means the person is not having any immune system. I mean, like immune system is, he lost his immune system. We know that. So they are prone to getting cancer. They are more susceptible to getting cancer. So that means somehow the immune system is protecting you. That is the inference you will have if you see like this. So these are all some of the examples. There is another beautiful example that came several years back. People found this, what do you see as the red colored ones here are the so-called tumor infiltrating lymphocytes. If you remember, I told you the so-called CTLs, cytotoxic T lymphocytes. What do? They kill the cancer cells. They help you. They prevent you from getting cancer. So they are good guys. Cytotoxic T lymphocytes. This tumor infiltrating lymphocytes are also like that. So people, pathologists, they found that there is a lot of this tumor infiltrating lymphocytes around the tumors. So another interesting thing is, whenever a patient is having a large number of these tumor infiltrating lymphocytes, their survival is better. That means it is having protective effect. Now, these are all the reasons for us to believe, these are all the evidences that convince us that immune system is indeed protecting us from getting cancer. See, otherwise, why the T lymphocytes, if they should be present in patients who are surviving better? Only reason should be these tumor lymphocytes, infiltrating lymphocytes must be protecting them. That is why they are able to have more survival. That is what is shown in this graph, which is called the Kaplan-Meier survival graph. You see this blue one, it means the patients having many tumor infiltrating lymphocytes. So their survival is better. They survive for many months, 100 months, 120 months, whatever. Whereas the people who are having few tumor lymphocytes around their cancer, they are dying out within 60 months. So that is what this graph shows. So these evidences convinced us that in tumors, immunology or immune system protects us from developing cancer. So if you understood that, that is enough. Now, one drawback, as I told you, for everything like the Newton's third law, we have something opposite. So the immune system also has certain breaks. This breaks means, what I mean is, checkpoint inhibitors. These are nothing but some molecules, some proteins, let us say. So they inhibit the immune system. If you inhibit the immune system, what will happen? The immune system may not be able to protect us. So they are sometimes called brakes. So if a vehicle, if you have a brake, you need the brake, because if it is going out of control, you want to control. So you need a brake. But in this case, if you put a brake, actually these are needed. That's why they are naturally present. Checkpoint inhibitors are there. They don't want the immune system to go out of control. I will tell you why you need checkpoint inhibitors in the normal system. I'll tell you that also. Because if you remember, what is the purpose of immunology? It has to recognize self from non-self or the foreign. It has to recognize only the foreign material and destroy it. It should not worry about the self molecules. Now, if there is no control on these, what will happen? It will start recognizing the self also. Then that is known as autoimmune disease. That's what is. Even your own molecules are recognized. What was thought to be normal molecule is now recognized as a foreign molecule, and it starts to destroy that. And that is why you get what is called autoimmune disease. So our own systems, our own body has certain checkpoint inhibitors. They act like brakes on the immune system. If you break that brake, you activate the immune system. Now, the whole Nobel Prize is about breaking these brakes or inhibiting these checkpoint inhibitors so that you can release the immune system to protect us. Because the immune system is unable to protect because of these checkpoint inhibitors. Now you break these brakes, and so that immune system is unleashed and our own body protects us from the cancer and treats us from not getting it aggravated further. That is what the Nobel Prize is. That is done by certain antibodies. That's what we are going to talk about. If you have not understood anything, if you understood this, that is the end of it. You don't have to worry. So who discovered this? How this immunotherapy came? James Allison at the University of Texas MD Anderson Cancer Center. Like Ram also mentioned, I did my postdoc in MD Anderson Cancer Center for about 2 to 3 years. I was there. But then I didn't work with James Allison. I worked with somebody else. He was a famous scientist at that time. So there was another scientist, Tasuko Honjo at the Kyoto University in Japan. Both of them won the 2018 Nobel Prize in Physiology and Medicine. They showed how proteins on immune cells can be used to manipulate the immune system so that it attacks the cancer cell. So these are the people. We will go to that. Now you may ask, sir, we know that cancer can be treated with standard therapies. We have the chemotherapy, we have radiation, we have surgery. So all these things are there. Why do you need immunotherapy? Well, I'll tell you one important thing. All the conventional therapies that I mentioned are good, but I must tell you, all the chemotherapy that we are using are highly poisonous materials. We are taking poisons knowingly, because then you may ask, sir, why knowingly, why doctors will give poison? It's not like that. I am just telling you for simplicity and for making you understand. We do that because they are deadly poisonous chemicals, and the drawback of that chemicals is that there is no chemical. I used to tell in the class to my students, if you discover a chemical which attacks only the cancer cell and not the normal cell, then you can be given Nobel Prize, because none of the chemical agents that are used to treat cancer now, they can be so specific to cancer cell and will not affect normal cells. It's not like that. In fact, they affect the normal cells, and that is what you call as side effects. Who does not know that cancer treatment has side effects? Everybody knows, because if you see that poor cancer patients, I happen to see the unfortunate patients when I was at MD Anderson Cancer Center. Whenever I travel through the hospital lift, I used to find children with almost like bald head because the hair would have fallen. Women also, women without hair, they may feel a little, this one very uncomfortable, lot uncomfortable, but then they cannot do anything. Once you give radiation, the hair falls. Not only that, that is a simple thing. Out of all the side effects, they get nausea, vomiting, a lot of pain, and several neurological sometimes disorder. In fact, I will tell you, the patients die sometimes because of the medication, also because the immune system is suppressed by these medications, and finally they may die out of what we call as opportunistic infections. You and I will not die if we get a cold or some virus, small viruses. Even COVID, many people are not dying actually. It's only a small percentage is dying. So as long as their immune system is okay, even you can be protected from COVID. But imagine these people who are given this treatment and their immune system is completely suppressed. That's all they die. In fact, many patients die like that. Many cancer patients also die because of what is called metastasis. Cancer has this property from jumping from one tissue. If you unfortunately have, let us say, breast cancer, you might get the treatment. The doctor says everything is okay, and after some 5 years, 10 years, you get brain cancer as a secondary cancer, or it may spread to lungs. This secondary cancer is this jumping property is what we call metastasis. More than 90% of the cancer patients die because of this metastasis. Now we'll come back to, I am distracting a little bit, but I'll tell you. So I told you the conventional treatments have certain problems. What are the problems? They lack specificity. They have side effects. Not only that, today you are giving some medicine, tomorrow you have to give tons of it to the same patient because he or she develops resistance. So that is another problem. So the resistance, relapse, it comes back again after 10 years. You thought he is completely remission, complete remission is there, and people are saying, doctors are saying, if you live for 5 years, you are okay, you are almost cured. And then one fine morning, you are getting it back. Relapse, they say. So the conventional therapies have these problems. So you need novel therapies. And for that, you must understand the molecular biology of cancer. Then you can understand, this is why people are saying, if you give a Saridan or some headache tablet or whatever, I can take one Saridan and my headache is gone, but you need maybe 2 or 3. So each person, it differs. This is what is called personalized medicine, they say. So each person is different. So also your molecular setup is, molecular profile when you get cancer is totally different. So if you can understand that, can we give specific therapy? That is known as targeted therapy. Molecularly targeted therapy. That is what is part of this immunotherapy also. So if you understand that, you can develop targeted therapies. Now I think I am distracting too much. We will go on to. So immunotherapy is one thing. It has the advantage of being specificity and remembering memory and then adaptability. So those are its advantages. Let us move on. Okay. I will just tell you, as I told you, these people got Nobel Prize because they discovered certain breaks in the immune system, and how to break that breaks is their contribution. One such break is called PD1. Just it's called programmed death 1. The other break that we are going to talk about is called CTLA4. I will tell you, it is cytotoxic T lymphocyte associated antigen 4. It's called. So these are the 2 breaks of the immune system. These people have successfully have broken these breaks so that they were able to enhance our immune system to protect us. This is how it was discovered. Now it was all in 1992, the cloning of PD1 took place. And then I will just point out the important discoveries. It was only in 2011, FDA, the Food and Drug Administration, approved one antibody for metastatic melanoma. I'm talking about this particular thing. And then only in 2015, the other antibodies were observed, and in 2014 also that the other PD-1 antibody, nivolumab, was approved. So it takes a lot of many number of years. Ponjo discovered T cell protein PD-1, which I told you acts as a brake in the immune system by a mechanism I will just tell you. It is not that these people are the first. Gordon Freeman, an immunologist at the Dana-Farber Cancer Institute in Boston, Massachusetts, along with immunologist Arlene Sharp at Harvard Medical School in Boston and Lee Shane at Yale University in New Haven, also studied checkpoint proteins, as well as a molecule that binds with PD-1 called PD-L1. L1 means ligand. This ligand binds with PD-1. Both are important. Both are acting as brakes. That's why I'm just telling about that. But PD-1 was only discovered by this Nobel laureate, not PD-L1. PD-L1 was discovered by somebody else. Okay. This is talking about the conventional immunotherapies. Immunotherapy, please do not think that antibodies are the only form of immunotherapy. There are a number of other forms of immunotherapy. You must have heard about CAR T cell therapy and cell activation therapy and so on. There are so many therapies, but each one has some advantages and disadvantages. Now, this is the most important slide. This is what we are going to talk about. This particular therapy, how it works. What is this therapy about? Now, I already told you that you have a T cell. Let us say you have a tumor cell. Tumor cell means cancer cell. So the T cell has this brake, immune system brake, what is called PD-1. And the tumor cells, the T cells also have the habit of presenting the antigens to the so-called T cell receptor. So the T cell receptors are present in the cancer cell. So you can see that the antigen is presented by the T cells, and that is received by the T cell receptors by the tumor cell. So why am I showing this? What is the connection, you can ask? Simple thing is, the question is how the immune system recognizes the tumor cell. How does it know it's a tumor cell but not a normal cell? I already told you that the normal cells have taken a different shape structurally. Some molecules have changed, and these molecules are now seen as foreign. Now, such tumor cells which contain these kind of molecules, cancer cells, they will have the T cell receptors. So the T cell receptors will recognize the antigen present in the T cell. This is how they get recognized. And also this PD-1, which is a brake. I'll talk about that. And PD-L1 is also another brake. So the tumor cell will have the PD-L1 as well as, whereas the T cell will have the PD-1. PD-1 is present in the T cell. PD ligand is present in the tumor cell. What happens is, if this ligand and the PD-1, if they interact, then the immune system cannot act. That is how they will put a brake. If this PD-1 is getting activated and binds with its PD ligand, then the immune system cannot function. That is how they put a brake. Now you understand the brake. That is, PD-1 is the brake. PD-1 is present in the T cell. T cell belongs to the immune system. Now this goes and binds with its ligand, which is present in the cancer cell. Once that happens, the cancer cells are happy. They will survive because the immune system can do nothing. In other words, cancer cells have developed a system by which they will interact with these brakes and will stop the immune system from acting on them. They won't break the brake. Don't get confused. They will interact with the brake so that the brake will not be they will be interacting with the brake, and so the immune system is paralyzed and it will not be able to act on the cancer cell. So it may be slightly confusing, but as we go along, I will try to make it more clear. What is this PD-1? PD-1 is programmed death. It's a just you can say it's a protein. It's a transmembrane protein. All these brakes that I am saying as brakes or checkpoint inhibitors, all these things we are saying, but they are basically proteins. So PD-1 is it contains 288 amino acids. It's a transmembrane protein. Let's not worry about too many technical things that are written here. These were prepared for some PhD students, so we need not worry about it. PD-1 is programmed death one. It's a protein, and it has to bind with its ligand, and then it will do what is called a signal transduction. It goes and binds with the cell and then initiates a series of events which we call as signal transduction. The PD ligand is again another kind of protein. It can bind with the PD-1. So that you have to understand. Again, it has got its own signal transduction pathway. We will come to that. Now, these people, these Nobel laureates, especially Honjo, they found out that PD-1 knockout mice, what it means, they have removed the PD-1 from the mice. PD-1, I told you, is a brake for the immune system. So if you remove that brake, the immune system becomes unleashed. It becomes very active, overactive. So what happens? You get some disorders. That's what is shown here. Arthritis, dilated cardiomyopathy, nephritis, myocarditis, autoimmune diseases. This is what will happen. I told you, if you release the over-releasing the immune system is also bad. When you don't have disease, when you have nothing to worry about, there must be some checkpoint. So don't get confused by that. But so these people have knocked out from the mice, and they found that the mice are getting. Why they have done this experiment? They wanted to understand what is the function of PD-1. They made a guess that PD-1 may be a brake. So they wanted to check that. So if you remove the brake, does the immune system get activated? That's what they checked. And they found, yes, the immune system got activated. That's why immune-related disorders came. That's what they found out. Now, this is a very important slide, but of course, a little bit difficult to understand, but I will just try to say as simple as possible. I will tell you how the molecular mechanism of this blockade. So what these Nobel laureates have done, they have put a brake on the brakes, and thereby they activated the immune system. So the immune system protected us from getting cancer. And this, they did it with antibodies. They developed some antibodies which could block these brakes. And that is how those antibodies are now in the clinics. They have been given to the patients, and the patients have recovered very well. That is why they got the Nobel Prize. But they were mainly working with this mice experiment that I was talking about, some of the cell lines that they were working with. So it was all basic research. But finally, it ended up in the clinic because they produced the antibodies, and they thought this antibody may be working with the patients because of their basic work that they have done in the laboratory. And finally, that proved to be true. And so they were rewarded with the Nobel Prize. This is the sequence of events. Okay. Now we will come to what is that they have discovered. Normally, see, the T cells have to be activated. You call it as what is called a T cell receptor. That is what is shown here. Normally, they will have what is called the T cells have to present the antigens to the T cell receptor. So that is what is shown here in the red color. This is called antigen presenting cells. So that is a signal number one. So antigen presenting cells have to present the antigen, and the T cell receptors will receive. Now, apart from this receptor, T cell receptor, there is what is called a co-receptor, which is called CDC28. This also should be present. and that will interact with the antigen presenting cells. There are other molecules which are called B7-1 and B7-2 and all that. This is the process to activate the T cells to protect us. I hope you understand. The T cells, what they will do? They will kill the cancer cells and will protect us. Or the cells that are going to become cancerous, they will kill them. So this is the process. So to do that, the T cells have to be activated. How they can be activated? They have to be activated like this. You have to present the antigen that has to be received by T cell receptor. And then there must be a co-receptor, which is called a CD28. And that should interact with the B7 and B7 point, whatever, B7, let us say B7 proteins. Now, once this happens, they get activated and they kill the cancer cells. Now, there is one enemy in the T cell receptor itself. That is what is called the break or immune checkpoint, which is given here. It is called CTLA4, which is present in the T cell receptor itself. In fact, it is a homologous molecule to the co-receptor CD28. I told you, whenever there is an activator, there will be an inhibitor. So the same cells, T cell receptors, contain activator CD28. They also contain a sister called CTLA4, which is actually inhibitor. So these two sisters, they fight with each other. Whoever wins will do. What the CTLA4 does, it will block this reaction. You see here, the CD28 interacts with B7. And now, instead of this, it's a competitive inhibition. Instead of CD28, now the CTLA4 comes here and binds with the B7. Now, the signal is attenuated. That means there is no T cell activation. The activation is closed. That is called attenuation. If you block the T cell activation, that is called attenuation. Now, for cancer therapy, you want activation, not attenuation. So what the Nobel laureates have done is, for example, James Allison discovered what is called anti-CTLA4 antibody. Now what will happen? This anti-CTLA4 antibody will block this fellow, CTLA4, I told you. This is the inhibitor. So it inhibits that inhibitor. It is a checkpoint inhibitor and it is inhibited by this antibody. Now the T cell gets activated and can kill the cancer cell. So we are activating the T cells, the so-called cytotoxic T lymphocytes, to kill the cancer cells. And so you are getting cured of cancer. This is one method. The other method Tasuku Honjo discovered is the PD1. Similar to this one, CTLA4, this PD1 is also present in the T cell receptor. Same mechanism. Now, this will interact with, see, this PD1, if it is free, then the cells can get activated. But if it is blocked by PDL1, then the cells cannot be activated. They will become attenuated. Similar to this one. Instead of CTLA, you have to imagine PD1. PD1 is another break. That break, if it interacts with the PDL1, then this will get attenuated. Now, you have to remove this attenuation. So Honjo came out with an antibody called anti-PD1 and he removed this interaction. Now the T cells are active and they killed the cancer cells. So this is it. The lecture is almost over. So this is what they have done. Now we are going to see what they have discovered and what patients they have used. Matter is over now. If you listen, I think my time is also over, it looks like, but I will take some 5-10 minutes. Is it okay? Tumors in PD1.
Please complete. Yeah.
Pardon?
Please go ahead, sir.
Okay. So the tumors in PD1 knockout versus normal mice, I just explained this experiment already, don't worry. So PD1 blockade inhibits metastasis. They found that metastasis, I told you, jumping up from one tissue to, see, all these experiments they have to do in animals, and then only it can come to humans. So they've done in vivo experiments, and in mouse models, they have shown it very effective. Then finally, they ended up with a human anti-PD-1 antibody, which is called, this name I want you to remember, it is called nivolumab, this antibody. There are many, you know, people remember there is some, it seems the people who had this cancer, they were dramatically, they recovered from taking this antibody. There is one famous actor in Malayalam, Mamta Mohandas or something like that. Yeah, she had some disorder. She was in California, and so she took this medicine, nivolumab, and she gave in an interview, which I happened to see, and she tells that she could immediately recover. So that is the power of these medicines. So it has become very popular, this nivolumab. I will, as we go along, we'll see. The other antibody, which I told you, James Allison discovered, CTLA4, that is called ipilimumab. These names are a little bit difficult. If you see this MAB means monoclonal antibody. If you see rituximab, something like that, XI means some recombinant protein, something like that. So the nomenclature is there. Whenever you see MAB, which means it is an antibody. Whereas if you see LIB, something like that, it used to be a chemical also. Sometimes they use chemicals, chemotherapy also. That will end up with IB, something like that. But if it ends with MAB, it's a monoclonal antibody. So there were a lot of clinical trials. They have used a lot of patients, 296 terminal stage patients recruited for 2 years. And they saw a very good response. 20 of 31 responses lasted for 1 year and more. Some even after stopping therapy, the people responded better. So that is how they became very popular. Similarly, this is not the simple thing. Anti-PD-1 is one thing. In ovarian cancer also, platinum resistant. See, the resistant cancers were cured by this antibody. Similarly, nivolumab, 3 milligram per kilogram, they have given. Lymph node metastasis also disappeared. Now, once this success story came, they started recognizing this therapy. FDA gave approval, and many cancers were given this therapy. Melanoma, lung cancer, renal cancer, Hodgkin's lymphoma, head and neck cancers, urothelial cancers, colorectal cancer, gastric cancer, the list goes on. Cervical cancer also, and so on. So many cancers were treated with this. The advantage is that it is less adverse effect. The side effects I told you about the conventional cancer treatment, little bit less in this, effective for a wide range of tumors. You may ask, sir, there is no side effect at all. No, that's not like that. There are some problems, and not all the patients recovered with this treatment also. Some patients were not responding. As I told you, each person is different. But many patients, they also combined this with, for example, in melanoma patients, they combined this with the dacarbazine, with the chemotherapy. So combination therapy, it is called. So that also they have done. And I will, yes, resistant tumors, they could, current issues. There are a lot of issues. Accessibility of killer T cells to tumor sites, potentiation of killer T cell function. People are trying to improve upon this. Only 30 to 50% of the patients remain unresponsive or less responsive PD-1 blockade therapy. So they are trying with, what do you do with these people? So you try with chemotherapy as a combination therapy, or you combine with some other chemicals, some other co-receptors, or 2-3 treatments together, or with mild radiation, and so on. Unfortunately, no synergistic effects have so far been reported. Let us not worry about this. Yeah. Treatment with the other chemicals also, Bisar fibrate and PD-1 blockade, enhances expression of genes. They found that certain genes are expressed once you give them together, which gave hope that this kind of treatments could be more useful. Combination studies underway. Future prospects, many more cancers may be treated with immunotherapy. Efficacy of PD-1 blockade therapy improved in 2016. 2020 also they are working many more cancers. Can we extend it to other cancers? They are working on that. Cancer may not completely disappear, but controlled by immunotherapy, cancer may become one of chronic diseases. Probably we will be at a point where we have discovered the cancer equivalent of penicillin. Somebody said that. Well, I hope cancer may be controlled by immunotherapy and its improvements. And they may also touch upon the microbiome. This is becoming a hot topic because we are living with more microbes inside our system. In fact, the number of microbes are more than the number of cells in our body. We have about 10 power 14 cells in the human body. We have much more than that, these microbes. That is called microbiome. And improving or tampering with those microbiome also can improve your health condition. I don't have time to talk about those things. We will almost stop here, and thank you for patient listening. I'm sorry I exceeded the time. I'll stop here.
No issues. Thank you, sir.
Sure.
Robin, did you collect any questions during registration?
Yes, that is question is there. But before that, there will be a flash quiz.
Yeah, Barney.
Yeah, that is Barney sir is there. I request Mr. Barney to take over the session. Barney sir.
Yeah, all the best, guys.
Okay, all the best.
Yeah, one second. I will open the session now. Okay, good evening, everybody. We are going to have a quick quiz on what has been discussed now. I'm going to paste a link. Please click this link. It will take you to a web page. Please remain on that web page. You will see 5 questions. We would like you to answer that question. So I hope by this time all of you are able to click that link. Please feel free to answer all the questions.
One of the questions I didn't give answer in the lecture, but let us see whether they are saying.
Okay.
Copy.
I hope you all have it now. The link is there, right? Okay, let me start the quiz. Yeah, quite a few have joined now. Let me give two more minutes. So that
Barney sir, I have pasted in the YouTube also.
Oh yeah, thank you. Yeah, anybody, both on YouTube as well as in Zoom meeting, please feel free to join. And 5 simple questions, you should be able to answer that and make it fast.
Finally, we had more than 500 people, right? 450
Yeah.
something. Yeah. Okay.
Okay, good. We will get started now. I'll start the quiz. The first question is on the screen now. Proliferating cells are always cancer cells. Is it true or false? It gives you 20 seconds to answer. I think that should be enough. Okay, a lot of people have chosen false. Let's see. That's the right answer. Is that right, sir?
Yes.
Okay, we'll go to the next one. Heart can get cancer. Again, true or false?
This is something I didn't give the answer, but let us see. I want people to wonder whether the heart can get cancer or not.
I don't know what we did. Okay, it's almost equal divided.
But yes.
Is it correct to say true?
Yeah, true is correct, but people have answered wrongly. That's
A lot
what
of
I
people have
expected.
chosen.
That's what I expected. That's why I didn't give the answer
Okay.
purposely. Because heart also can get cancer, but it's very rare, very, very rare. But it can.
Immunotherapy is specific to only one type of cancer. Is it true or false? Okay. False is the right answer.
Okay. Correct.
Okay. Cancer is an infectious disease. True or false? Okay, many of you chosen false. Okay, good. The right answer. The last question. Most of the cancer patients die because of metastasis. Okay, let's see what is the right answer. True. True is the right answer. Okay, that's it. Who is the leader? Oh, many people have got 5 out of 5. Fantastic. Congratulations. The fastest finger is Sidra. They made it first.
Wow.
Congratulations, everybody. Robin, I hope you have noted down. We will. Yeah.
Yes, hardest question because the answer was not given.
Yeah. Okay. Thank you.
Thank you, Mr. Bharani.
I will put a most interesting question, perhaps not hardest.
So we have collected the questions from the registration form itself. I'll share the questions, sir. You just go through the question and answer.
Choose your questions.
Yes.
What is it?
We have, while registering, we had got some questions from students.
So what should I do? What should I do?
So if you want, I can read up these questions, sir.
No. I can read out. I can read. That's not an issue.
You can pick up which one.
Is this similar to immunotherapy? What do you mean by is this similar? This is immunotherapy.
This is immunotherapy. Yeah.
Does it have negative side effects, or what do you think will be the patient's life experience? All these things I have answered in the
covered. Don't pick up such questions which you've already answered.
Yeah. Which cancers do not respond well to checkpoint inhibitors? What drugs are used for immunotherapy? Yeah, these are all the things that they are working on. Many cancers they are trying to work on that I have mentioned in the slides. Yeah. What cancers are working and what more cancers have to be done. Everything was shown in this one. So I will try to see whether something.
Yeah, quickly if it is something it's not been answered.
Though there are vast research taking place in the cure of cancer and immunotherapy, there has been significant cure found in cancer. Terminal stage, why is it? Okay. One thing is there is always this cancer, this intriguing feelings are there. Why it is not getting cured or something like that. Problem is it is only from your genes. As I told you, it's not alien genes. It is your own genes. So when you touch your own system, there is bound to be, even practically speaking, when you see a cancer, the surgeon or chemotherapy or anything, it is difficult to distinguish between cancer cells and normal cells because some cancer cells are embedded between the normal cells. So how do you specifically remove them? So that becomes a very challenging question for even the surgeon and for other people. Even practically, I am saying, although I'm not a doctor or something, but this is one problem. Molecular biology wise, because our genes are affected, normal genes are affected, so it's becoming difficult to cure it that way when it becomes a terminal kind of thing. Yeah. Of course, if it is discovered before, we can do it. Yeah. Is that what is that? Proteogenomic studies involve cancer treatment? Can you say anything related? I don't know what is the proteogenomic studies. Yes, proteomic studies are being done, though there are vast research taking place in the cure. Okay, that we have studied in the permanent cure for cancer. We can inject resist Covaxin, Covishield. Yeah. The vaccination is there for certain virally induced cancers like Covishield. We have, I'm forgetting the name, cervical cancer, we have the vaccine. So those virally caused cancers, you can have shield like Covishield. You can have, yes. That is for prevention. Don't confuse with the treatment. Which is more effective immunotherapy, T-cell immunomodulation, the macrophage immunomodulation. These things, don't worry about that. The people are working on that. Is liver cancer curable with this T-cell immunomodulator? I'm not sure about hepatocellular. I think yes, it is also one of the cancers, but I cannot be sure. I'm not a doctor after all. Our body also has immunity to prevent the tumor in body, but how the cancer tumor phytoproteins present, they escape from the immune system. There are many mechanisms that I have told some mechanisms, but you cannot explain everything. There are maybe hundreds of mechanisms. At least some 10-15 mechanisms are listed in the textbook. So we cannot be discussing all the mechanisms. It will be difficult. How can we reduce the effects of tumor cells once detected? There are a lot of diagnostic methodologies. For each cancer, there are different types of methodologies to detect the tumor cells. For some of them, they use the imaging system. For some of them, they have certain other tests. Virally caused cancers, you can detect through some other, some ELISA-based methods. So many methods are there. Yeah. What cancers are treated with immune checkpoint? That has been told. Okay.
That has been listed already.
Yeah.
Last slide.
Yeah. Next.
There was another question in the chat box.
Yeah.
Yes, chat boxes.
Where is it?
Shall I read it, sir?
Read it out, no?
Yeah, read it.
Just 1 minute. Let me see. Which
It
one?
says, can we, sir, why does passive smokers become more prone to lung cancer? Meanwhile, more than the active smokers.
Yes, this is one question I used to discuss in the class also. I will, see, the problem is this is not a full lecture on cancer biology. On lectures in cancer biology, I used to tell this point. It's very simple. I'll explain it again. All of you or many people wonder why somebody you know who has been smoking for 20 years did not get cancer. Why someone who started smoking yesterday, so to say last year, he started smoking and he gets cancer. This is one intriguing thinking among many people. People think like that. Although I cannot pinpointedly answer that, I will tell you there are 6 hallmarks for cancer people have discovered. I cannot describe them fully because it will take a longer time, but just understand there are more and more mutations have to happen. Let us say you are going to reach some destination. You have to cross 10 stations. Okay. Let us think that these are different stages of cancer. So somebody is standing, somebody, if he smokes, that is one stage. Somebody drinks, that is another stage. Somebody doesn't work, he is very lazy, that is another stage. So there are many factors, risk factors that are there. Somebody gets infected or somebody gets exposed to radiation, that is one stage. So supposing one person is in the 9th station, now he starts smoking, it only takes one more station for him to reach the 10th station and he reaches the destination. So the person who got cancer by smoking, by starting to smoke last year, he is already having so many defects in his system. Now this has added as an additional point and now he got cancer. Whereas the other person who had all these smoking habits for several years, he needs some more aberrations for him to get cancer, because cancer requires at least 10 to 15 mutations, a minimum, to get a full-fledged cancer. I have simplified the answer. But
I thought the question was about passive smokers, like who happen to be in the vicinity of the smokers are affected more.
Oh, was it?
Well,
okay.
That was a question.
Okay, this is one question that I told. The passive smoking, yes, that is because passive smoking also, first of all, each individual is different. Your susceptibility depends upon your own genetic makeup. So that is one kind of answer. The other answer is passive smoking. Also, the smoke gets into your lung, and if your lung is not capable of taking that or rectifying that, then you will immediately get affected. How you get affected depends upon your sensitivity.
Your response system to that.
Yes, your response system to that. Yeah. Then.
So can we cure cancer cells by targeting their genome and by gene therapy?
Yeah, people are trying. You see, this is such general things, each cancer is different. So we have to know which cancer you are talking about. Even among the same, let us say colorectal cancer, even among the colorectal cancers, each person is different. So we will have to look at their molecular makeup and try to devise a particular targeted therapy. That is why it's not easy task. Yeah.
And another thing is from Mohana Priya. Is cancer genetic and contagious?
Contagious, you have yourself answered that it's not an infectious disease.
Not an infectious disease.
You answered it.
Yeah.
So that question is not
null and void. Yeah.
Is it genetic? See, people get confused with the genetic and hereditary. Hereditary means if I have, my son also will have, something like generation to generation, it is fine. Genetic means it is related to genes. So cancer is genetic, yes, because it is related to genes. It is not hereditary. Hereditary is only a small portion.
Different.
Yeah, hereditary is different from genetic. Genetic simply means genes. So genes are connected. Yes, it is a genetic disease. And also you have epigenetics nowadays. What is called epigenetic factors are there. That's a long story. Yeah.
If chemical content in our food is high, then is it true that chance of getting cancer is more?
Yes, carcinogens do cause cancer. Not only in food, in the environment, whatever. You will have the first chemical carcinogen being a cancer-causing agent was discovered in what is called the soot chimney sweets, as they call. That is in England. In those days, the 7 to 8 year old boys were employed to clean the chimney sweeps, because the sweeps will have a small hole. You and I cannot get into that. Boys are so lean, so they can go. At that time, they won't wear any dress. And so these boys will get into that naked, and their scrotum was affected and they got scrotal cancer. That was the first evidence for any chemical causing cancer. It was in 1765, something like that. Percival Pott was the discoverer. It's somewhere in the 18th century.
Now, this is some technical question, I guess. I'm not able to read it. So can we recognize and clear cancer cells using CRISPR-Cas mechanism?
CRISPR-Cas mechanism is basically to delete some genes. So that mechanism can be used to study cancer. Yes.
Sir, would our immune cells eliminate the monoclonal antibodies that we use as therapeutic, so that our therapy may be affected?
They may not eliminate. In fact, the antibodies will try to do something with the these things. They may not eliminate as such, but there are some other antibodies that could be developed and that may defeat our this thing. That is anti-antibodies could also be developed. Yeah.
do metastatic cancer cells undergo a period of dormancy?
Yes, yes, yes.
And foods are the main reason for causing cancer. Are foods the main?
No, no, there are many reasons. Food is also one of them.
Again, the same Hariharan is asking, how does a cancer kill the patient?
With one word, I cannot answer that. There are several mechanisms. Of course, one thing is the failure of many mechanisms that are there as a protective mechanism. One mechanism is what we have discussed now. If it suppresses the immune system, then you get killed. That is one mechanism.
So, permanent solution for cancer, we have to hope that it becomes a chronic disease by 2050, 2030. Somebody has asked permanent solution to cancer.
It is like
Ongoing fight.
taking some measures like being active in life and not leading a sedentary life, and checking yourself regularly. For example, women can check up both their breasts as well as the cervix. Regular checking should be done, so we can almost eliminate it. Many cancers, of course. Some cancers are curable also.
Will chemotherapy affect negatively?
Chemotherapy will definitely affect. That's what I told you. No, if you discover a chemical that won't affect normal cells, then you can be given Nobel Prize. I told you. What more effectively you can say that?
Can you tell us more about the heart cancer? Which therapy is used to cure it?
No. See, heart cancer, I just asked for this thing. Heart cancer, you don't normally detect it. What happens is the symptoms are very similar to heart attack. So you don't detect it. It comes as a passing this one. When they are doing it for other forms of heart defects, they also discover the tumor, but mostly they will remove that part, surgery or something only they will do.
Can cancer be passed from the mother to the fetus during pregnancy?
Yes, if it is hereditary.
If it is. Can immunotherapy cure cancer caused by a mutation in the DNA repair genes?
Yeah, that is what it is.
How to spot early stage of cancer?
Yeah, there are methods. Diagnostic methods are there. So one has to go through that. For example, cervical cancer means papillary smear is there, pap smear is there. Yeah, pap smear is there. Breast cancer means mammogram is there. So like that. Yeah.
Does epigenetics play a role in cancer?
Just now I told you, epigenetics is a vast topic. It plays a tremendous role. That's why when somebody asked genetic disease, I also told epigenetics.
Genetics.
Yes, both are important.
So,
both the environment and the genes are important for causing cancer. Yeah. Yeah.
So the brain cancer happens in neuron.
Yeah. That is a relevant question with heart cancer also. Does the brain get cancer? Because brain cells are not dividing. Why are they getting cancer? Because the concept is only the dividing cells get cancer. Like, when you are walking and if you are stuck with a stone, you may escape with a small injury. But if you are running, if you are stuck with the same stone, you may even die, because the speed with which you are walking will have more injuries to you. Similarly, proliferating cells will get cancer, but not the undividing cells, but not the cells which are slowly growing. So in that context, why brain cells are getting cancer? Because they don't divide, they don't grow well, and so on. That is a very good question. The answer to that is the brain cells, what we are colloquially saying as brain cells, are not really the neuronal cells. We are talking about what are known as the auxiliary cells, like glioma cells, schwannoma cells, and so on. So they are in the periphery and they are growing well. Actually, they can grow well. Astrocytes, Schwann cells, glial cells. So accordingly, you get glioma, schwannoma, astrocytoma, and all that. In rare cases like heart cancer, neuronal cells also can get cancer.
Sir, one question I think she has missed. So what is the recent type of cancer spread nowadays?
I don't know. You'll have to look at it.
Okay.
Altered gene function and malignant cellular transformation is called epigenetics. And
No, epigenetics, if you want to define that, there are certain changes which are inherited by you. In this case, you are inheriting that change, but the genes do not change. You have inherited some genes, but their gene sequence do not change. What changes is their expression. That is called epigenetic change. Basically, epigenetic means apart from genetics, because genetics cannot explain everything. There are a lot of things that I can tell about that. See, what happens is when a human is born, how it is happening by the fusion of sperm and ovum, and you call that a zygote. Zygote is, it looks like one cell, right? Zygote, because there was one cell, sperm cell, there was one ovum, that is another cell, but both the cells are have fused, and you get what is called a zygote. This looks like only one cell. Now, if you look at that zygote, it looks like a cell and a mesh. But when you look at the embryo, then you start seeing eyes, nose, some ear-like thing, and even some tail-like thing, everything you are seeing. If you see a rat embryo and a human embryo, they will pretty much look the same. But finally, when they come out, rat is a rat, a human is a human, and you know. Now, what you have to wonder is how this zygote, which looks like a cell mass, how it has become something like an embryo which has some shape. This is called differentiation. Now, if you take each cell, the genes are same, whether it is embryo, whether it is a liver cell, heart cell, or brain cell, everything. The genome, I told you, about 20 to 30,000 genes are there in each cell. So the genetic content is same, whether it is a liver cell, heart cell. But we all know that a heart cell is a heart cell, liver cell is a liver cell. It functions differently. Why? Because all the 20,000 genes are not kind of expressed. They are expressed differentially. Some genes are expressed in some tissue. That's why liver cell is a liver cell. Some cells are expressed in brain cell. That's why brain cell is a brain cell. This is why in stem cells, from one cell, from a fibroblast cell, you can even get a brain cell, because basically the genes are same. So if you tinker with that, you may even differentiate that into a brain cell. So this is gene expression is important. Epigenetics plays with gene expression. So if gene expression is tinkered, then you can change the whole thing. That is the idea. That is why epigenetics is very important. That is, in other words, you can convert the normal cell into a cancer cell if you play with gene expression.
Can cancer recur after immunotherapy is one question.
In most cases, no, but in some cases, as I told you, it's only 40 to 50 responders, are for 30 to 50% do not respond, they say. In some cases, they might have found. I don't know. This is one of the latest things. And as I told you, I'm not an immunotherapist. So many people, I only know about these discoveries, but about patients and patient-specific questions, I should not be able to. I won't be able to answer. Yeah.
My school friend was affected by brain cancer. She lost her past memories, and she died in a few months.
Yes.
Is this possible to lose memory?
Everything is possible if you touch which part of the brain you touch. Brain cancer in the the fourth grade, if you have, their days are numbered, only 2 years. There is no cure for that. So it is a pathetic thing. Yeah, brain cancer, especially if it happened to be a metastatic cancer, because brain is a notorious metastatic site, which means secondary site, which means the person already had some other cancer.
Primary somewhere.
Yes, correct.
Can malignant tumor cause removal of organs? Is it possible to lead a normal life once the tumor is removed?
That's what I'm asking. You are asking like asking a doctor. I'm not a doctor.
I'm not a doctor.
But it should be possible, yes. Many cases.
Unless it is not a secondary. If you remove the primary from the ovary, I have seen people surviving for quite some time.
Yeah. See, I am a molecular biologist. Any questions with relation to genes and other things, what is happening? But this fundamental, that's what in the beginning of the lecture I told. Fundamental science and application, both are important. Without this foundation, that doesn't come. I am more on the fundamentals. If you touch fundamentals, I may be okay. But I become shaky if you touch the other thing because I do not know. If you ask me what is the medicine given to treat breast cancer, I will say nothing. I don't even know the name of the medicine sometimes. I mean, some of them I know, but not everything. Because I am not an oncologist. There are divisions even there. Nowadays it's becoming like that. An eye doctor doesn't know anything about nose, right?
Right nostril and left nostril have two doctors.
Yeah, two doctors. So even among the cancer doctors, there is a medical oncologist, surgical oncologist, and so on. So if you ask a surgical oncologist about something about the medicine, he may not be telling you fully anything. He will say, you ask my friend who is a medical oncologist.
One last question is on, is there immunotherapy for lung cancer?
I think I listed that.
And is radiation required or not?
Radiation can be given in combination. That also I told. They are trying wherever they are not very effective. They are trying combination therapy, either radiation or chemotherapy or whatnot. Whatever you see, you are hopeless. So you try everything. Yeah.
So I think we will call it a day today.
Yes.
Robin?
I think yes.
Yeah. Thank you so much, Professor and Ram, for bringing such an excellent person to start this theory to practice session for the year. I'm very happy that we connected with Professor Karnakaran of IITM Biotechnology department. I forget the big name that you gave there, but anyway. And I hope every one of us who have been here would have enjoyed the session, and it required a very deep engagement with the speaker because I couldn't switch off and do something else. I was taking notes like in a class and it was very, very engaging, and I really am very jealous of the students of yours who had been in your class actually. So that is even though it's a new subject for me, we are familiar with cancer right from film to the recent times of COVID. Every other person is having one immunoglobulin and this and that, and it's a very costly medicine. Those are the things that we keep hearing. And we hope and pray, as Professor said, it becomes a chronic disease quickly like any other disease in this world, and it becomes treatable. And thank you. Every participant here had put in their best in being engaged with this lecture. I thank each and every one of you and for making this lecture extremely useful for yourselves and for us who had been trying hard to give the best lectures to you. I thank Professor once again. I thank my colleague Ram for bringing this person once again, and all the audience who are here and in the YouTube. And thank you, PMO, for putting up a great show. Thank you very much. And you need to attend take the feedback and take the test, which is given there to be eligible for certificates. So I will expect all of you here to take the test as well. Thank you very much. Thank you.
Participants, I request to submit the feedback as well as online quiz.
Yes, stay back.
Are posted in the Zoom chat box as well as the YouTube.
In fact, I would like to remark for the benefit of the audience that actually you asked so many questions, but what you should know is my cousin is an oncologist. So he says from the US, first you go to a clinical oncologist, then he will decide what it is. Then only he will send you to a, whether it's a radiation oncologist or this thing.
Yeah.
So here directly you go to a radiation person and he does
Actually advises radiation.
allowed in the US, you know. So it's very, very specialized. So that is something I want the students to know, that the subject is very vast.
Yeah, the treatment part is very difficult to
even the doctors are divided.
Yeah.
And Here, sometimes you go direct to a doctor, just like you go to a physician, family doctor, only then you have to go to a specialist. So that way it is practiced nowadays. So that is something which I just wanted to share. Yeah.
The family physician will naturally know your genetics. You'll say, "Unga mama ka so you may also be suffering with this." My father was a general practitioner. So I have seen him.
That generation is gone.
That generation is gone, yeah.
Yeah. Now he doesn't know you also. Okay.
Don't have to, because with all the scientific interventions, like it's not just the x-rays today. So many interventions. It's more engineering than medicine.
Technology has helped. So you have so many methods. And the one thing is cervical cancer used to be very heavily affecting the US women in those days. But now it's not there. So the screening procedures have helped. So there are
cervical cancer. Women who get more children or more engaged in those activities get cervical cancer. If they don't feed their children properly, they get breast cancer.
No, that is about
one way or the other. We are due for a cancer at some point of time. It's just that we have to be aware of when it is happening. So periodic checkups will help for any women. That's about all. We have to get it in the early stages and then address it. That's all.
I told about that.
Yeah.
Thank you.
Okay.
Thank you, Professor.
So I can leave, right?
Yeah, thank you, sir.
Okay, thank you.
Thank you.
Thank you.
Thank you, Ram.
Lalitha, thanks.
Thank you.
Thank you. Yeah, Robin, can you close it, or you're waiting